Today, we have a very exciting guest lecturer, Martin Hamburg, who will be teaching single-cell RNA-seq analysis both for today and for Thursday. So Martin has a very interesting background in physics, in chemistry, in bioinformatics, and also in economics. And he was a postdoc at Harvard Children's Hospital from 2007 to 2014 and has been a group leader at Sanger Wellcome Trust, but recently returned to Harvard Medical School and the Brigham Women's Hospital to be a faculty. And he has been a pioneer and expert in single-cell RNA-seq analysis, having authored a very influential paper in Nature Review Genetics on single-cell analysis. So today, it's our great pleasure to have Martin teach us how to do single-cell RNA-seq analysis.
Thank you very much for the introduction, Shirley, and thanks for inviting me to give these two lectures here. It's a very interesting and exciting field, as you probably don't need to hear from me. And as Shirley mentioned, I've pretty much been working in this field since about 2014 when I started my own research group. And the field has really grown exponentially in that time. So there's a lot has happened, and today it's sort of impossible to cover everything in just two-hour-long lectures. So this is really going to be a bit of a whistle-stop tour, and I'm only going to be able to sort of touch upon the most important findings. And I hope that one of the things that you'll excuse me for is that there will be a bit of a bias towards the methods and the tools that I have been part in developing, because those are the ones that I know the best. So that's my declaration of this not being completely unbiased overview. So anyway, so the first question is sort of, why are we doing single-cell RNA-seq? Well, so traditionally people have been doing bulk RNA-seq, and the sort of what you do there is that you put a lot of cells together, which sort of initially people did just because they needed to sort of have enough of transcripts to be able to get to be able to carry out these profilings. But there are sort of course, major drawbacks with this. The sort of most obvious one is that if you have a complex tissue, then you'll be mixing all the cell types that you had in there together, and so you'll get like an average profile. And this is, of course, very problematic if you have complex tissues, such as the brain, where you have multiple different types of neurons and also non-neuronal cells, or even if you're looking at many other tissues, you'll have, for example, you'll have resident immune cells, which will be different from the fibroblasts and the stromal cells and the epithelial cells that you have there. So sort of merging all those together and just getting an average profile, you miss a lot of information. But what you also miss is that we know that there is a high degree of variability between cells, and that type of profile is also lost. So you can't tell how much two neighboring cells are differing in terms of their gene expression levels. So this, of course, people knew that when they started doing bulk RNA-seq, but it took a lot of sort of technological advances in order to be able to sort of make these technologies work for a single cell. And now there is a sort of well-established single cell analysis workflow, which sort of pretty much starts with the sort of you have to start by dissociating your cells, so you get sort of single individual cells that you can profile. And these this can be sort of, for some tissues, for example, if you have blood, that this is trivial, whereas for other tissues, as for example, the brain or the heart, this can be very challenging, and you have to put a lot of effort into sort of optimizing your protocols so you don't damage or stress the cells too much. But after that point, it's pretty much you have the same workflow as you would have for normal RNA-seq. The main difference, of course, being that you start from much smaller amounts of materials, so you have to be very careful so you avoid biases. We're not going to be talking very much. We're going to about the sort of the experimental protocol here. We're covering a little bit about that, but most of the efforts here today and on Thursday will be about the sort of what you do with your data, your sequencing data, once it comes off the machine. And again, conceptually, it's the same as for bulk RNA-seq, but there are some some important differences, and what I'm going to argue is that you basically need sort of specialized methods to be able to get the most out of single-cell RNA-seq data. You can't just use plain old bulk RNA-seq methods. So, so as I mentioned, that one of the real advantages of single-cell RNA-seq is that you're able to profile a sort of large number of cells, and you can get the entire transcriptome. So that allows you to really sort of to quantify very well how big a difference there are between neighboring cells. So there were sort of really pioneering studies sort of done in the 90s and the noughties where people were able to sort of see that there is a huge degree of noise or variability between different cells. But this, of course, was only done with sort of one or a handful of genes, but now you can do that for the entire transcriptome instead, which is can give us a big insights into how this variability is regulated and what roles it plays. But what is perhaps even more exciting is the fact that we can profile these individual cells. And since the cell really is the fundamental building block of biology, this allows us to really understand some complex tissues from a sort of bottom-up point of view. And this insight led a sort of a large number of researchers to get together in around 2016 to form this International Human Cell Atlas Consortium, which has a very lofty ambition, which is to sort of profile every cell in the human body. And the goal here, you can really think of this as a sort of intellectual follower to the Human Genome Project. So the goal is to build this resource of profiles of every tissue in the human body that sort of other types of research can be built on, and so that's what they're hoping to achieve here. And this idea of building a cell atlas is of course not unique to human biology. So this concept of atlases has been sort of caught on by many different other fields. So not surprisingly, there are several efforts out there to build a mouse cell atlas, and they're arguably the ones that have come furthest. There is also an effort in the Drosophila community to build a fly cell atlas, and there are also more exotic atlases. So, for example, the malaria cell atlas, which is interesting in the sense that it's the first single cell organism that has been profiled. So its entire life cycle has been profiled, and this is actually not as easy as you might think, because it has a very complex life cycle, and obtaining samples from all the stages is a very complex endeavor. So before we sort of start diving into everything about single-cell RNA-seq, I just want to sort of have a few words of warning and say that it's sort of, although it has been shown that if you take enough cells from your single-cell RNA-seq experiment and then sort of aggregate them, it will sort of start to asymptotically to look like your bulk RNA-seq experiment. So that's obviously nice. It sort of shows that we really are building up towards bulk. So that might lead you to think that single-cell RNA-seq is always going to be better. But I don't think that's really the right conclusion to draw, because for one thing, it's more difficult to do the experiments, and it's also more expensive, and you really have to ask yourself, sort of, do we really need this information? So, and I think that sort of bulk RNA-seq is going to be around for quite some time, and there are definitely uses for it, but we'll not be talking more about that today. Today, we're going to be focusing on the single-cell RNA-seq stuff. So here is a very nice overview of how the field has grown, and as you can see here, we have a log scale on the y-axis and a straight line, which basically means that we have an exponential increase in the amount of cells that people have been able to profile as they have been reporting in the literature. And the field, the birth of the field was really in 2009, when there was a paper from Azim Surani's lab, where they profiled 8 mouse embryonic stem cells. And so that was the very first single-cell RNA-seq paper. As you can see, we have had the growth. And now you would see that sort of the typical paper will have on the order of 10 to the 4 to 10 to the 5 cells that they report. So that's I would say it's about an order of magnitude increase every 2 or 3 years. So what this really has been driven by is a technological development. So as you can see here from the colors in this, so these are the same dots just colored by the technology that was used. You can see that there have been sort of several waves here in terms of different technologies that have dominated what people have been using. And another way of looking at the field, which also shows you an exponential growth, is just the number of publications that come out with single-cell RNA-seq. As you can see here, we still have an exponential growth. And what's interesting, if you break it down by what technology they were using, you can sort of see that we have the Chromium, 10x Chromium technology, which has sort of really overtaken the field, sort of much like a SARS-CoV-2 variant can spread in the population. And this really is the dominating technology. And it wasn't really, very few people would have heard of it five years ago. So it really shows you how quickly these things can change. So yeah, so I think this is the end of the first part. So I will stop the recording here. If I can figure out how to do that. Right. Yeah, so there's a lot of technological development going on in this field, and it's moving very, very quickly. And but I will give you a brief overview of some of the technologies that are dominant today, as well as some of the ones that have been most important historically. Oops. So SmartSeq2 protocol was developed in 2013 by Richard Sandberg's lab at Karolinska, and it is what is known as a sort of full-length transcript protocol. And so, which means that it allows you to profile reads from the entire transcript, which is sometimes very advantageous, in particular if you're interested in studying isoforms or splicing. It has a very high sensitivity, and it's also relatively low throughput. And today, it really is the most popular protocol from the sort of relatively low throughput protocols. One of the disadvantages of this protocol is that it doesn't have unique molecular identifiers, or UMIs, which we'll be talking about a little bit later. There was, however, recently published another protocol from the same lab, SmartSeq3, which does incorporate UMIs. However, I think it's a little bit too early to tell if it is going to be taking off and become as popular as the SmartSeq2 method. Another method in the sort of low throughput, high sensitivity category is CellSeq2. From Itai and I's lab, and it was really developed with high sensitivity in mind. Unlike SmartSeq2, it does not do full-length coverage, but it will only give you the 3' end of each transcript when you profile it. So a really landmark in this field was in 2016, when there were 2 papers published in the same issue of Cell. Interestingly, both of them from labs in the Longwood area. So we have the DropSeq, which was from Steve McCarroll's lab by Makosko et al., and the InDrop, which was from Alan Klein. And they're both based on the same idea, which is that you basically encapsulate each cell in a little droplet, and then you carry out the lysis of the cells inside that droplet, and then you sort of attach small barcodes to all the transcripts that you extracted, and then you can pool all the RNAs that you extracted together, and then you can sequence them, and then you can later, thanks to the barcode, find out which cells they came from. So this really upped up the throughput, and it was a major breakthrough. So, however, what is the type of droplet-based method that is by far the most popular, as I mentioned before, is the commercial one, which is called the Chromium from 10x Genomics. And my view on this is really that the sort of the InDrop and the DropSeq platforms, in terms of the performance, and the cost, I think they're relatively similar. In terms of performance, they're relatively similar. I think that what the 10X have done really, really well is that they've done the engineering really well. So I think that you pay money to them and then it basically works. Whereas the InDrop and the DropSeq has the advantage that you can customize it and sort of set it up more to your own liking, but it requires a lot more work and a lot more expertise. The other thing from which is important to know about the 10X platform is that it comes with this software called CellRanger, which you'll probably come across quite a lot, which is sort of their platform for analyzing the data that comes off the machine. So another method related to the droplet-based techniques is called single-cell combinatorial indexing, which was developed by the Shandura Lab at University of Washington. So they are sort of trying to sort of deal with this issue that where you have some of these cellular barcodes, which is something again we'll talk about a little later, can be quite short. So what they are trying to do essentially is a combinatorial strategy where they put cells together with these barcodes, so that each cell. So a number of different cells will get sort of similar barcodes, and then they just mix around the cells again, so that they end up in different compartments, and then they get assigned a second barcode. So the probability of two cells getting assigned the same pair of barcodes is very small. So you basically have a very few collisions, and that really expands the number of cells that you can profile using a relatively small set of barcodes. So this is a very clever trick that they have been using to do this. Another method, which I think is worth mentioning, is called Sequel, which was developed by the Sharlach Lab at MIT. So here, the basic idea, right, is that you want to put each cell in a small volume and then carry out being able to carry out these reactions. But what they're using instead of using droplets is that they're using microwells. And so you basically wash the cells out into the microwells at sort of very diluted concentration. So you get only one cell per microwell. And then you can sort of seal it as sort of illustrated here. You can carry out these lysis and capturing, and then you can pool it and you can sequence it just as before. And really the main advantage and the main selling point of this method is that it doesn't require the same type of kit that you would require for these droplet-based methods, where you need to have this sort of microfluidics apparatus. So here is the hardware that you need to do these experiments, which makes this method really portable and very suitable for field work. So I think again it's important to say that I don't think there is sort of you can't say that there is such a thing as a single best method. All these methods here are complementary and they'll have different advantages. So when you're deciding on which one to use, you really need to think through sort of what is your biological question and what are the things that are most important to you, including budget, of course. And I think there are some sort of important or sort of major patterns to point out here. So one is that you really have the sort of one division is between the sort of more high throughput versus the technologies, which are the droplet-based methods versus slightly more low throughput methods. And the cost of having high throughput, at least for now, but this of course may change in the future, is that you lose quite a lot of sensitivity. So what that means is that if you take the sort of the same cell type and then sequence them to the same depth, you will detect fewer genes in these droplet-based methods. And initially, these droplet-based methods were sort of restricted to only sequencing the 3' end of mRNAs, but now they've changed their chemistry so they can also profile the 5' ends, which typically gives you more information. So that's usually better to do. So I think, so in addition to doing these sort of qualitative sort of evaluations of which technology to use, you can also do more quantitative benchmarks. And if you look into the literature, there will be sort of several papers that are doing this. And these papers are very important because sort of a lot of labs will be looking at them and trying to decide which technology to use. And they also need to be done quite frequently because there is such rapid innovation in the field. So every 2 to 3 years sounds about right to me. So here is a recent such paper, and here is the kind of setup that they're considering. So they're considering these 7 different methods here, and they group them into these 2 groups, the low throughput ones and the high throughput ones. And then they do 3 types of experiments. So the first is a sort of human and cell line mixture. So this is very simple. You put together some cells from a mouse cell line and from a human cell line. So the advantage of this is that you really have a sort of a very solid ground truth to evaluate against, because you're basically trying to decide if you have a human or a mouse cell. The second type of samples that they're analyzing here is human PBMCs, which is nice because it's a very well studied type of system, so it's sort of as close to a ground truth as we could come. And then finally look at some samples from mouse cortex, which is again a very well studied system, but also much more complex. So this gives you sort of evaluation for these methods in different types of settings. And the type of things that you can evaluate is sort of shown here. One of the figures is, for example, the sensitivity. So how many genes do you detect when you sort of sequence to similar depth? And as you can see here, these low throughput methods come out as having higher sensitivity, and of the high throughput methods, you can see that it's the 10x chromium that comes out best, which is sort of might explain be one of the reasons why it's so popular also. So yes, I will stop here. Right. So yeah. So for the rest of these two seminars, we'll be talking about the computational analysis of this type of data that comes out, the data that you get off your sequencing machine. And the first part is really about pre-processing and quality control. And if you ever worked with genomic data before, you'll know that it can be very noisy. So this part is actually very important. And if you don't get this right, there's a very good chance that you will be analyzing sort of technical noise and other types of artifacts and mistaking those for real biological signal. So fortunately, at this stage, there is the general workflow for how to process this data is very well established. So here is a very nice overview from a recent review. So as I told you, that sort of the first part is really about pre-processing and doing various types of filterings and normalizations. And then sort of the second part is really what you refer to sort of more biological type of analysis, where you do the type of study where you can sort of try to draw biological conclusions and inferences. So yeah, so the first thing you need to do is really just to look at your reads and do quality control of those. So this is no different from any other sequencing experiments. So you can use whatever type of favorite tool you have for that. I'm not going to go into too much detail, but I'll just show you sort of one small example here from a study which I was involved in. And so what you can see here is that you have, this is an output from FastQC, which is a popular tool. So you can see here is that you have sort of somewhat lower quality at the beginning of the read and at the end of the read, which is sort of fairly common. What's surprising here is that we have this unexplained dip right at the middle of the reads. We have no idea where it came from. In this case, we sort of decided that it's unlikely that this is going to have a major impact on the mapping or anything else. So we just decided to keep all the reads, but this is sort of important to look at in order to make sure that you're happy. Then of course there can be various types of adapters and so on that they have put on for the experiment. So you need to be able to trim those off as well. And there are also softwares for doing that. So the next step that you need to do is demultiplexing. So that's basically identifying which read came from which cell. So if you're using one of these sort of low throughput experiment where you typically put, like for example, SMART-Seq2, where you put the cells in different wells here, then you would also know sort of which barcode for each cell that was put in each well. For the droplet-based methods, this can be more complicated. So you might have to do a little bit more work for the demultiplexing. And it should be said that in sort of, if there had been no errors, this would of course have been straightforward in principle. But what we also have to take into consideration is that we can have sequencing errors on these barcodes, and we need to account for those when we do the demultiplexing. So here is a sort of an overview of the type of a schematic overview what you'd get out from your experiment once you've done the demultiplexing. So this is for a droplet-based type of experiment where you'd also have this unique molecular identifier, the UMI. So here we've been able to group each read by the cell, which was this barcode that was unique to each cell, and then we have this UMI. So now I'm going to tell you next about what the UMI are. So UMI stands for unique molecular identifier. And it's basically a short tag that you assign, you attach to each of the transcripts prior to amplification. So this allows you to sort of distinguish the reads that came from, so if they came from the same molecule, the same initial transcript, then they will have identical UMIs. But if they came from different transcripts, then they will be different. So this is a way of sort of very efficiently removing PCR duplicates, but it also allows you to sort of do absolute quantification, because you know that each UMI will correspond to one transcript. So here is sort of illustrated how useful these can be, and this is a very small schematic example. So here, for example, the red read got sort of amplified 4 times. And so if we then went ahead and did sort of variant calling here, you might assume that you would have 4 of the A's versus 2 T's, whereas in reality it should actually be only 1 A and 2 T's here. And that conclusion you'll be able to get if you have the UMIs and if you collapse the reads that corresponded to the same UMI. So UMIs can lead to trouble again, because you can have various types of errors. So one error that can happen is if you have very abundant transcripts, and you'll often have quite short UMIs that can be as short as 6 base pairs, for example. Then there is a risk that you'll have collisions. So you'll have initially it was actually different transcripts, but because the pool of UMIs was relatively small, they will end up getting the same UMI, and then you will think that they were PCR duplicates when they weren't. Another issue is of course that you can have sequencing errors in your UMIs. So then you might think that things that were really different, things that actually came from the same transcript actually instead are different because of these errors. And you can, but fortunately there are many different tools available for dealing with these types of problems. One of them is called UMI tools, and it is based on using a sort of a graph approach. So basically what they do here is that they look at sort of nearest neighbors in terms of the number of errors or differences in the Hamming distance of these UMIs. And then based on the patterns, they can sort of infer what types of errors were the most likely to have occurred. So here is a fairly common example, a fairly common situation, which is when you have one node which has a very high number of counts. So that's presumed then to be the sort of the original originating UMI, and then you'll have that surrounded by nodes with much smaller counts, and that is then likely to have been caused by having at some point during the amplification process you had an error, and hence you have fewer reads mapping to those UMIs. So if you take this into consideration, then you can sort of prune this and basically merge the UMIs that were sort of caused by this error process, and so that will allow you to avoid those types of errors. So once we've sort of collapsed our UMIs, and so then we need to sort of align the reads and quantify them. And here there are really sort of 2 main strategies. The first one is to sort of use the traditional route, where you would use a mapper such as STAR to align the reads to the transcriptome, and then you can use for example, feature counts or HTC to just quantify the number of reads that you have for each gene. The other method, which is very popular when you have these very large data sets, is to use a pseudo-aligner instead, which, for example, Salmon and Callisto are the two popular ones. And I would say that that's probably the most popular strategy these days. There are also methods that can sort of are specifically designed for these droplet-based experiments to do the UMI analysis and the quantification. So Alevin is one such method from Rob Patro's lab. So so they really sort of try to model the process of these droplet-based methods and sort of try to do clever things in order to reduce the errors from this UMI deduplication and the quantification. And this should hopefully lead them, allow them to sort of rescue some reads that would obviously have otherwise would have been hard to map, for example, due to multi-mapping. So this is sort of illustrated here in this figure from their paper, where they compare against CellRanger. And as you can see here from their genes where reads are uniquely mappable, you don't really have much of a difference. The difference really happens here towards the left end, where you have these reads, genes that are sort of probably arose due to duplication events. So there are a lot of reads that are areas that are multi-mapping, and here their method is able to do much better. So presumably you get better quantification of these genes, whereas here it doesn't matter very much. So now that we've sort of understood what our UMIs are doing, and we've counted those and sort of mapped to the reads, to the genome, then we can sort of start quantifying the different genes. So now I've extended this schematic that we had here before, so we can also sort of see how many counts we have for each different gene in each of these different cells. And so the next step to do it requires us to take a step back, and this is again really specific for the droplet-based methods, and to sort of consider an important aspect of the sort of experimental design here, or how you carry out the experiment. So remember, the goal is to put one cell into each droplet, and this is, of course, the cells are very small, and so are the droplets. So this is not as trivial as you might think. And what happens in practice is that you end up with a Poisson distribution of the number of cells in each droplet. So you want to sort of, so now you need to strike a balance that on the one hand, you can think that's basically something you control when you put your cells into the machine. The number of cells controls the rate of this Poisson parameter. And if you put too few cells into the machine, then you have a very low rate. So the problem then is that you'll end up with a lot of empty droplets, and that is basically a waste of these reagents. So that means that you'll get sort of fewer cells out of your experiment, and it will cost more. The other problem is the other direction is not good either, right? Because then you put in more cells, and then you end up getting more doublets, and those can really sort of screw up your analysis, because it's not obvious a priori if you had one or more cells inside a droplet. So we need to strike a balance here, and of course we need computational tools in order to be able to identify these problems of empty droplets or droplets containing multiple cells. So this is what I'm going to be talking about next. So the way you can think about this empty droplet detection sort of conceptually is very straightforward. So basically you just count the number of unique UMIs that you found in each droplet, and then you'll rank them. And what you're really looking for is some sort of a big drop where you, and the hypothesis then is that big drop would correspond to a situation where you go from having viable cells with UMIs, to having these empty cells where you just have sort of various type of debris that somehow got encapsulated. So this is a very typical situation, that you'll have a sort of a few cells with a lot of UMIs in them. And that those might correspond to doublets, and then you'll have a sort of a long range here, and then you'll have some sort of drop here at the end. And this is sort of this drop where you need to sort of decide where to do your cutoff. So the most straightforward way is just to sort of do a simple thresholding, There are, of course, more sophisticated methods for doing that. One example is this empty drops method, which sort of uses it like a Dirichlet model to try to build a model of the sort of the background distribution, and that allows them to sort of look at the contents of each cell or each droplet and sort of compare it to this presumed background. And if they look very different, then they can sort of rescue the cell and not throw it away. So that allows them to keep some of these cells that have lower counts, whereas the threshold method would just sort of drawn a line here and thrown away everything to the left. So that is hopefully a little bit better. The other problem that I was telling you about is sort of doublet detection, and this is where these dual species experiments that I talked about earlier are very useful. When you do those types of experiments, you're hoping for this type of plot, which is shown here, which is known as a barnyard plot. So, but what you're basically hoping is that you would get one cell in each droplet, and then you can map those reads that you get from each droplet to both the human and the mouse genome. And then you would, of course, hope that you get a lot of matches to one of the genomes and no matches to the other. And this is sort of what you see here when you have the points sort of hugging the x and the y axis. But then, of course, you have a few points here on the diagonal, and those would presumably corresponds to these multiplets, where you have sort of one cell from human and one cell from mouse. So this type of experiment you'll often see when people develop new technologies, and it's considered an important benchmark. Now, of course, in most practical experiments, you won't have two species to deal with, but you'll only have cells from one species. So then you'll need a different type of approach. And again, there are multiple methods to do this. Two of them are called Doublet Finder and Scrublet, and those and others, they use pretty much the same strategy, which is basically that you take your cells, and again, you assume that the number of doublets is very small. And so what you do then is that you create artificial doublets. So you take two cells and you create the doublet, and then you analyze this sort of new sample where you have introduced these artificial doublets, right? Because you know which ones they are. And what Doublet Finder does is that it then builds the nearest neighbor graph, and then it takes the cells from the original sample that look most similar to these artificial doublets. It will assume that those are actual doublets as well, and then it will sort of throw those away. So this might sound like a bit of a circular reasoning, but it seems to work quite well in practice. So another type of problem which is sort of related to this is called ambient RNA. So ambient RNA is basically you'll also hear the term the soup. So that's RNA that somehow gets into all of the droplets. So it has presumably leaked from some cells that were in your samples, and then they've been able to enter all of the droplets. And now they will sort of contaminate the samples by sort of raising the background levels across the board. So this is obviously something that you want to get rid of, so that you don't think that you have these transcripts everywhere, because it can mess up with your differential expression analysis. So the first method that tried to correct for this background and identify the ambient RNA is called SoupX. And what they do is that they look at the very far end of this tail here of the droplets that contain the fewest UMIs, and then they assume that corresponds to the soup, and then they sort of build a model of the soup, and then they try to sort of subtract it from the other cells. I think the main drawback of this method is that it requires you to input a list of genes that should not be present in each of the cell types that you've found. So that sort of can really raise the bar in making this more complicated to apply this method. So there are other methods out there. I think the most recent one, which a lot of people seem to be using these days, is called CellBender. So this is based on neural networks. It uses a deep autoencoder. It basically allows it to it will build a model of both of the the noise and of the signal, so it can detect both ambient RNA and doublets. And as you can see here, it seems to be doing a really nice job of sort of basically removing these types of sort of low-level background reads, and to push the background almost all the way down to 0. So so now basically what we've done is that we've from our droplets, we've removed the ones that were empty, we've removed the ones that were doublets or corresponded to multiple cells, and we've also removed this sort of background soup. So now we hopefully only have sort of droplets left with individual cells in them, but we still need to do another round of filtering. And it's basically to try to sort of remove low quality cells. So this, what people typically do here is that they look at sort of 3 quantities. So one being just the number of reads, the other one being the number of genes detected, and the third one being the fraction of mitochondrial fraction of reads mapping to the mitochondrial genome. So the reason for looking at the last bit is because it's basically if cells are sort of dying, then they will typically release a lot of mitochondrial DNA. So this is really an indicator of a cell which is sort of stressed or is problematic in various ways. Unfortunately, it's sort of hard to, there are no sort of strict rules for what is sort of considered an outlier in terms of the number of genes or the number of reads. It's really something which depends on your sort of on your platform, on your sample, and so on. And so there are no hard rules. Really, what you have to do is it's more of an art than a science, and you have to sort of, the goal is to sort of remove outliers, and you're looking for things that are sort of reasonably Gaussian. And so that's what you really need to strive for here to get the nice uniform shapes. And again, you want to remove on both ends, not just at the bottom end, but also if you have outliers with very high counts, that they can also mess up your analysis later on. So yes, I think I will pause it here. Right. So I think an important word to be said is about this, all this pre-processing and quality control business. I think is that my advice is always to sort of, it's always painful to sort of throw away data, but my recommendation is to be on the sort of stricter side and sort of throw out things that are dubious, because you don't want to. We know that we have lots of artifacts and lots of noise, and you don't want to sort of end up having that confounding your analysis. And if you think that you've been too harsh, you can go back and sort of try to rescue some of that stuff. But if you're going to err on one side, I would be on the sort of more aggressive side in terms of the filtering. So yeah, so now we're basically gotten to the stage where we can build something called an expression matrix or a count matrix, which is what is sort of really the central object in the sort of computational analysis from single cell RNA-seq data. And it's sort of what most of these methods that we'll be talking about later for doing the biological analysis are sort of based on. So it's an important concept. So by definition, the expression matrix is really just a gene by cell matrix. So each row corresponds to a gene, and each column corresponds to a cell. And if you ever looked at one of these matrices, what you'll know, the most sort of conspicuous feature of it is that they tend to be very, very sparse. So they have lots of 0 entries. Much more so compared to what you'd see from your bulk RNA. So here, this example that I'm showing you here is from a SMART-seq2 experiment. So it's actually relatively dense, but it's not uncommon to sort of see from these really high throughput experiments that you'll have 90% or even 95%. I think the highest I've seen is like 98 or 99% zeros. So they can be really, really sparse. So now, this sparsity is of course, there's a reason for it. So one of the reasons is that it could be that you have just sequenced at a very low depth. So you're not processing the transcripts that were in there. Another reason is that you might sort of see having these rows of zeros. So those would presumably correspond to genes that are absent, sort of in that cell type. So and again, you would expect to see more of those when you have single cell compared to when you have bulk, where you sort of merged everything together. But this is sort of not enough to explain this high degree of sparsity that we're seeing, because what you'll also find is that you'll find some rows where you have some values that are non-zero and can be relatively high, sort of interspersed with these values that are zero. And if you look even more closely, you can see that this can happen sometimes for genes that you'd really think would be like markers for the cell type that you're looking at, but they seem to be completely absent. So these are often referred to as dropouts, and it's sort of thought that they are mainly a technical artifact, that for some reason the transcripts from that cell were basically lost, or the RT reaction failed, or somewhere along the application they didn't get along, or it was just sort of low sequencing depth that prohibited you from seeing those. So what that basically means is that when you see a zero in the expression matrix, it means that you can't really know for sure if it is really a zero and that represents no expression, or if it is a missing value. They're both recorded as the same thing. So that is conceptually a problem. And then, of course, having lots of zeros in your matrix is also problematic from a sort of mathematical or statistical point of view. They will cause lots of problems. So so the next question I might want to ask yourself about is, so if you have a given gene, what kind of distribution does it follow? Because we think of this as the outcome of a stochastic process. So since these are count data, you might think that it's Poisson, but it's of course much more has much wider distribution. So the more accurate or the better description is instead a negative binomial, which again, the negative binomial can emerge from a sort of Poisson mixture model. So basically, if you assume that the rate for the Poisson process is instead of being constant, is a gamma distributed random variable, then you end up with a negative binomial. So I think that's a sort of useful way of thinking about where this negative binomial comes from. And initially, people sort of saw these zeros and they started thinking that there was an excess of zeros. So people would use sort of the zero inflated negative binomial. But now I think the consensus in the field is that it's actually not, the number of zeros is actually what you would expect. So you don't need to have zero inflation to sort of have a good fit to the data, but just a plain negative binomial should be sufficient. So the next thing that you want to do here with your matrix is normalization. So this is something that we're familiar with from bulk RNA, and it's really to compensate for these issues that can that come from having different sequencing depth in each of the cells. But of course, it could also be that you just had different RNA contents in your cell, which will make it more complicated. So people have thought quite a lot about these types of normalization methods. So some of the methods that were used in bulk RNA-seq will work really well, some won't. So for example, the quantile normalization or the size factor methods tend not to work very well because you have so many zeros, so that's why they will fail. So RPKM you typically don't use it if you only have the tags, but the UMIs allow you to actually do sort of counts per millions or transcripts per million, so TPM or CPM. So those methods tend to work really well. There are, of course, methods that were sort of specifically developed for doing normalization for single-cell RNA-seq. One of the first one was SCRAN, which basically sort of takes a size factor approach. But in order to sort of avoid this problem with zeros, what it does is that it creates pools of cells. So then you'll have much fewer cells. So it creates lots of these pools. And then it can sort of calculate size factors per pool. And then it can deconvolute those pools to get the size factor for individual cells. Another method is called SC-norm, which basically takes a sort of regression type of approach. So it basically divides the genes into 10 different deciles, and then it sort of corrects for each of those quantiles separately, and it sort of tries to achieve normalization that way. The most popular method these days, I would say, is SC transform. So it also uses these type of regression approach, but it does so in a sort of more sophisticated manner. It uses sort of negative binomial regression, and then it looks at the residuals from that. And so it does that. It does that quite successfully, sort of remove the dependence on the expression level of the gene, and it also has some sort of impressive performance because one of the problems that Hafemeister and Satyja identified is that if you do this normalization, that what you'll often end up is sort of inflating the variance. So it manages to do this sort of compensating for the sequencing depth and do variance stabilization at the same time. So that's sort of illustrated here in this synthetic example where they took some cells and then they took just half of them, the cells, and downsampled them by 50%. And but this method basically is able to compensate for that, and it finds sort of, as you can see in the UMAP, where the cells are sort of nicely and evenly spread out, and it doesn't really find any differentially expressed genes between these two groups. So another type of issue that we need to correct for is what's called experimental batch. So batch correction, which is again something that you'll hear a lot about in the single cell world. So these batch errors basically occur when you have sort of consistent sort of technical problems. So you can have, for example, if you have sort of experiments being done on different days, or different people running the experiments, or in different labs, or different batches of reagents and so on, that can introduce these sort of technical differences that sort of makes two samples that really were the same look different. So the best way of getting rid of these experimental batches is sort of through proper experimental design, so that you can basically mix your sort of biological conditions across different replicates, and then you will be able to sort of identify what were the batch issues and what was the real biological signal. Now, this is of course not always possible. For example, sometimes we have samples that need to be processed right away, and then it's impossible to do this proper experimental design. But fortunately, there are some computational methods that allows us to sort of rescue this later on. So one thing that's important to keep in mind about these methods is that which is not always obvious when you look at them, is that they're sort of operating in different spaces. So ideally, you would really want your method to operate sort of on the expression matrix. So then you will basically you feed your method the expression matrix, and it gives you a corrected method expression matrix. And then you can do everything that you wanted to do with it. But some of these other methods that are out there, they operate in sort of in other spaces, and that means that you can't really do the same thing with your matrix once it's been corrected as you would have otherwise. So that's important to keep in mind. So the most popular method historically has really been COMBAT, which was actually designed for microarrays. It's really robust. It's sort of based on a sort of linear regression type of approach, where you sort of assign each sample a sort of a batch variable, and then you can regress that out. So despite the fact that it uses a straightforward approach and it sort of assumes normality and so on, it seems to work remarkably well. So it has really it's really aged well, this method. There are more sophisticated methods that typically are more computationally costly. One of the very popular ones is called Harmony. So they sort of take a sort of soft clustering type of approach, and they sort of try to sort of find these sort of centroids that sort of where the clusters will have can have sort of multiple memberships, and then they can sort of use that to sort of infer how the cells should go together, and it also allows them to have like sort of partial memberships that can be helpful. Another very popular method is from this Seurat package, which we'll be talking about later, and you might already have heard of. They sort of use sort of align the two different samples using a sort of canonical correlation analysis type of approach. And then they use a sort of mutual nearest neighbor strategy, which is sort of similar to nearest neighbor, but for the two cells to be mutual nearest neighbors, they both need to think that the other one is their closest neighbor. And so that allows them to sort of identify sort of so-called anchors. And then based on the differences between these anchors, you can basically calculate a correction vector that you can apply to your sample, and that allows you to sort of get a corrected matrix. One of the things that's very important about these corrections, so both the normalization but also more importantly the batch correction, is that can actually sort of distort the expression matrix. So especially the batch correction, what they are really designed to do, these methods, is to sort of make sure that sort of cells that are actually sort of the same cell type, but because of these technical artifacts sort of end up a little bit apart from one another in the expression space, are sort of assigned to the same cluster. So they will sort of put these two clusters together. And when they also modify the expression matrix, they may not sort of care what the method is doing to the expression matrix, as long as it gets the cells that belong together. And that can be problematic for sort of when you're doing other types of analysis. So here is an example where we looked at just marker genes, so genes that are sort of highly expressed in the different clusters. And what you can see here is that these different methods, they can actually change the profiles of these the sets of marker genes that you get out quite significantly. So whenever you're doing these batch corrections, it's actually very important to sort of also do the analysis in the sort of in the uncorrected space to make sure that you're not sort of being misled by these corrected matrices. And so the final topic I'll talk about today is imputation. So as I mentioned, we have this problem with all these zeros, and the idea behind imputation is very straightforward. So we can use some sort of mathematical model to try to guess what should have been instead of the zero. And that will basically get rid of the problem of having a sparse matrix, because now we can sort of populate it with non-zero elements. So the first method to do this is called magic, and it is sort of based on a kind of diffusion type of approach. So essentially what it does is that it looks like a sort of nearest neighbor type of graph, and then it's sort of the values from non-zero values are sort of basically allowed to diffuse out and sort of spread into its neighbors. So as you can see here, it does a really good job at sort of densifying this expression matrix. So and it sort of fills out these blanks here really, so you can get sort of much nicer continuous trajectories, and you really don't have any issues with the zeros anymore. What I should sort of confess here is my bias, that we put out this study a couple of years ago where we looked at a number of these different imputation methods that sort of were becoming quite popular at the time. And one of the problems that they have is that they weren't the only way they were validating their methods was by doing sort of positive controls. They weren't really doing any negative controls. And what really happens with these imputation methods is that they create false positives. So what we did was a very simple experiment, was that we took the original expression matrix, we shuffled it, so there was absolutely no structure in terms of the expression profiles, and then we searched for these differentially expressed genes using like a very conservative methods. And of course, what happens with these imputation methods was that they sort of found these patterns that really didn't exist, and then they sort of amplified them, and that leads to sort of very high false positive rates. So this is this is problematic, and it's sort of, I think right now imputation methods aren't very widely used, and people are quite wary of them for these reasons. And there are, of course, people trying to deal with this. So I think the most productive approach is to basically use an external reference. So this is where you take advantage of these different atlases, and the idea here is really conceptually is sort of clear why it could work, that if you take sort of outside information and you add some information, you're not just sort of smoothing out the data or guessing what it would be based on your model. So these I think have a better chance of working, but they're still not sort of, I wouldn't say that they're widely in used. So yeah, so this pretty much takes us back to the roadmap that we had here in terms of what the analysis can do. So now I think we've pretty much gotten to the bottom of the first column here. So we'll be talking about some feature selection and some dimensionality reduction and visualization next time, before we go onto this downstream analysis. So with that, I will stop sharing and I will stop the recording.